Hey guys, welcome to our last topic in Chapter 6. How does microbial growth work? I know we've been overwhelming you with vocabulary in the last two topics, so this one, hopefully you'll be able to take a little bit of a brain break in the vocab section and put a little bit of work towards applying some of the concepts we've been working on this chapter. This is a short topic, but once again, same reason it's important before. If we know how microbes grow and what their growth rates are, we can control them and use them for laboratory work. Or if we want to stop them, we can stop the growth within those environments. So now let's talk about how cells divide. It's critical if we're going to talk about cell growth to know how they grow, right? And microbes, especially bacteria, use binary fission. They divide, or I guess it's They divide with bacteria will divide with binary fission. They're the only ones that do that. Eukaryotes use mitosis. So bacteria in bacterial binary fission, one bacteria cell becomes two. So it's very similar to a mitotic process, except for in this case, it's more basic because there's less involved in this. The most important part of binary fission is that the genetic information and the ribosomes and everything else get copied. Otherwise, when the cell splits, we're going to have one cell with half the information, one cell with the other half. So it's really, really important that we understand how those two copy. And we're not going to go into that in this chapter. In this topic, what we're looking for is growth. So rate of bacterial growth. There's a couple important terms. I know I said there wasn't going to be much vocabulary, but there is a little bit of vocabulary, just a little bit. So a generation is the time that it takes for a population to increase by a factor of 2. And generation time or doubling time is the time required for the complete fission cycle. Okay, so make sure you understand those two concepts. Bacterial growth always occurs exponentially. You can see that on the chart on the top with that red line is the growth. And this is because we start with one cell, we go to two cells go to four cells, cells go to 8 cells, etc. So it doubles every time. And so that makes sense that it would start off slow because the numbers are low, but then once you reach a certain amount, it's going to go crazy. And you can see kind of an example of that on the bottom picture here. So it's really important to understand that this is an exponential growth, and it's always an exponential growth. The exponential growth may be a different time point, but they're always in a constant rate depending on the microbe that we're talking about. So this exponential growth is part of the overall growth curve. And you can see exponential growth phase here in the second spot. So first we have lag phase in the growth curve. In lag phase, the bacteria is getting used to its new environment. There's only a few cells, so it's not rapidly dividing. It's really, really slow. Okay, then we had that exponential growth phase that we were talking about, and this will continue for as long as you have plenty of nutrients and environmental space. Okay, so as long as it's not too crowded, you'll keep going. So if you take a small inoculum into a large volume of media, your exponential growth phase is going to last longer than if you take a large inoculum and put it into a small amount of media. This is why in lab we tell you to just take a little bit, because then it'll keep your cultures in the growth phase when you come back in 2 days, as opposed to taking a big old swipe from your previous inoculum. We then reach the stationary phase. This is where nutrients are starting to run out, waste products are starting to build. It's kind of like in a big city, right? There's not tons of space. And so what happens is some of the microbes are going to begin dying, and they're going to die at the rate the new cells are being made. And so that's why it flattens out. And this is your stationary phase. This will last for as long as it can until the waste builds up to such a place that the death phase begins. In the death phase, more cells start dying than dividing. Now, you can catch this to a point where some cells will always remain viable. There should never be a place where the entire culture dies unless you've done something to it. But it's important to be able to catch it there. Now, when we work in the laboratory, we always want to catch our cultures in the exponential growth phase. Bacteria behave more typically when they're in the growth phase than when they are in the stationary or the death phase. This is why we try to have you inoculate little bits so we can do it like that. If they're in the death phase, some of their characteristics may be different. They may not behave normally. So it's really important to try to always catch them in the growth phase. And as you can see here on this table, it shows that the time is much smaller than what is our normal what we do in lab, where we have 48 hours, but that's because it's just a condensed version, and this curve will vary depending on the cell type. For instance, E. coli doubles rapidly, and so their growth curve is a lot more condensed than some that grow more slowly. Lastly, let's talk about how we calculate growth. It's important if we're growing to know how many cells we have. And there are three different ways that we do this. The first is with turbidity, and you can see that in the bottom picture here. In the bottom picture, what we use is a machine, and we will shine light through it. And that will tell us how much light is being transmitted and basically how turbid it is, how cloudy it is. If it's really turbid, then we can get that measurement. If it's less turbid, we can get that measurement. And then we will apply it to some known standards and that allows us to determine the turbidity or the cell growth based on those calculations. Turbidity is quick and simple. It's an easy way to do it as long as you have the previous calculations already worked out. So if you're just looking for something fast, turbidity is the way to go. We can then count, and this process gets a lot more complicated and takes a lot more time. We can do serial dilutions like you guys have done in lab. We can also do direct counts, and we can also use flow cytometers to do the count. So there's a variety of different ways, but they're all more cumbersome than just the turbidity. But it gives you a much more accurate count. You know pretty much exactly where you're falling. The last is genetic probing. This is a lot more involved process, a lot more expensive. In this case, we use RNA to determine how many cells are present. So it's really accurate. It's also really expensive. So that's where we go from here. So that's why it's turbidity is the easiest and the cheapest, to genetic probing is the most expensive and to an extent time consuming. So that's the three different areas. So make sure you understand how that works. And I know this topic was really short, but we're at the end, so you get a little bit of a break from it after we blew your minds in the last two topics. So make sure you review those objectives and let me know if you have any questions. Growth curves are really important, so make sure you fully understand exactly what's happening in each of those phases. And let me know if you have any questions.